Hey guys, what is going on? It's Alexander Williamson, and welcome to Fishtory, episode 124. So, today we're going to be talking about some newly discovered Rasboras. So, in the last few years, there have actually been quite a few Rasboras discovered, and some of them have been discovered in Malaysia. Two have been discovered in India, and then several have been discovered in Borneo, in the Sarawak province, and also in Indonesia in the Aceh province. There have also been a couple discovered. So out of the total 150 species that have been discovered, there are 39 species that are distributed across Borneo alone. And there are 6 in the Sarawak region of there alone beyond that. So there are a total of 8 different Rasbora groups, and this was all kind of sorted out in 1984 by an ichthyologist with the last name of Britann. And those groups are Trifasciata, Argrotinea, Enthenovinii, man, I'm bad at these, Daniconius, Laterastratia, and Caudule maculata, as well as Sumatra elegans and Pauci perforata. Man, those are hard to say for me for some reason this morning. But anyways, those are the 8 groups of them. Now, one of the problems they've had is that they look really similar sometimes. They're all small fish, so they're kind of hard to figure out what species are which. And in a lot of cases, they're in water that is really shallow, really tannic, looks like Coca-Cola. And so it's difficult out in the field to figure out when you have a new species, even if you're an expert. So even ichthyologists are having a hard time. But one of the new fish that was discovered is the Rasbora notura. And it was discovered by Maurice Cotelat. And they also have discovered a number of other danios and sunda danios, as well as just a number of other cyprinids in Southeast Asia. So in the Journal of Ichthyology Exploration, they have some new articles that have come out, and they said that the existing Rasbora nutura has been confused with the Rasbora bungarensis, and that the Rasbora nutura has quite distinctive coloration, with a black stripe running from the start of its dorsal fin to the caudal fin base, and a large diamond-shaped mark on its caudal peduncle. So even though it has that, some of the other Rasboras, including this burgundensis one, has similar markings with a dot instead of a diamond, and a line that doesn't go quite as far. So they thought it was just a morph of the two, which brings up the whole point of what's going on here, and what this paper is talking about, and that is that it's really hard to tell them apart. So now they're getting into genomics, and they're using samples of these fish, scale clippings, just take a little scale off of them, or if they pass away, they can use a specimen too. But then they are able to run the genes and sequence the genome and see what is what. Now, what's interesting is that there's also another species that was discovered called Puntis shola, which is found all the way over in India. And it has a strong and rigid last undivided dorsal fin ray, whereas the others are divided, and it is similar to the Puntius dorsalis, which was discovered in 1849, and to the Nigronatus plamatulil, the Nigronatus nelsoni, and the Plamatul Verdis plamatulio, Gosh, guys, that's all Latin to me. They don't have common names yet, but these are existing fish that are just seen as kind of boring and not colorful. And so, morphologically, there hasn't been a need for the industry of selling fish or food fish to figure out which of these are their own species, which of them have been double named. Because sometimes you'll think, oh, two phenotypes, two different looking fish, two species, and then you run the genome and you find out they're the same fish. So what they have done just now with the advent of these new species being discovered, and people saying that there's Rasboras all the way over in India, as well as all the way down into Borneo and Papua New Guinea, is they're saying, let's whittle down this catch-all term of Rasbora, because it really just means a specific group of cyprinids, of Cyprinidae. It's a subgroup, and it's kind of a catch-all for things like Danio is, or Danionella is. And so now they're going through with 103 different collection points of what they think are the existing species, which was really cool that somebody went to all that trouble to go collect every single known species. And then with cytochrome oxidase 1 inference and genetic testing, they're then creating a morphological identification. So they're making each morph its own ID, and they're trying to then decipher which are their own species. So it is interesting in that one species in India, like I mentioned, could be it could be 4 different species, or it could all be the same species, or it could be 2 species of Rasbora, 2 that happen to be really just 1, so a total of 3. We don't know yet, and so they haven't posted the findings of this paper, but it'll be interesting. And to get more of the Sarawak region kind of discovered and on paper and research will be a really good thing, especially because they've built the Bakun Dam there, and they wanted to reveal the impact on fish diversity of the Rasbora caudimaculata and the Rasbora borneensis, among others. The poor water quality and stagnation up in the reservoir of the dam is probably causing some problems to the 35 species that are only found in that water. So out of all the Rasboras in the world, 23.8% are found in that one river, behind that one dam, or in the tributaries thereof, and are endemic to Borneo, whereas 14 of them are found elsewhere and also are in Borneo. So in the Centarum National Park, they are trying to create kind of a safeguard for these species, and hopefully in that national park they can get some IUCN listings, and rather than threatened with threatened species status currently, which 10 species have, they're thinking more like 24 need that status, because a lot of them now they're finding, oh, that's its own species, and it only occurs in one river, and that river's being polluted as we speak. So this work that they're doing right now, which of course I'll report on when the final papers are published in the next year or so, will specify whether the little Rasbora that's found in a peat moss bog and that's found in a raging river that look identical are the same. Now, one interesting thing to come out of this set of papers is that they said the best way to actually tell if a species is in fact its own species is to look at the distance between the dorsal fin and the tail, and to measure that distance or that ratio in comparison with the head and the actual tail, and that will tell you more reliably than any other feature that they've discovered so far if it is in fact just a different phenotype, a different morph of the same fish, or a whole new fish. That being said, there's probably another 30%, if not all the way upwards, some people say up to 100% more of these fish that we need to discover deep in the jungles of Southeast Asia. There are probably by some accounts, there's another 100 to discover. Other people say no, it's probably more like 30 or 40 more species that are out there. But nobody really knows for sure. You could find one lake where there's 50 species altogether. So it's important that we keep looking and keep studying these creatures, because then we find out new and interesting things about how they work, where they live, and how they evolved. And sometimes that can apply to human uses, from medicine to industrial and material design, all the way to physics and all sorts of other things. So that is the update on your Rasbora news for now. There's links in my information where you can go there and you can look up pictures of some of these new fish, but I can't share them as they're copyrighted right now and just were published like a matter of days ago, and in one case a matter of a year ago, in scholarly journals, which are a little Sue happy when it comes to sharing their stuff without permission. So sorry I can't show you guys the cool Rasboras. Actually, they're all fairly plain Rasboras, if I'm being quite honest, but they are always interesting to me, and I always like seeing new fish regardless of how vivid they are in multicolors or what. Alright guys, that's it. This was a long one. The rest are going to be shorter this week. I appreciate it. Appreciate you guys hanging in there too. It's been a long week for me. I lost my cat of 12 years, and we had to put him down yesterday, and he's been sick for the last week, and I've been to the vet 4 or 5 times in the last 9 days. So just having a go of it, but I wanted to make sure to get this stuff up and out. And thank you all so much for supporting the channel and what it is I do here. See you tomorrow on The Secret History Living in Your Aquarium, featuring the Fishtory show. On the Aquatic Morning Show too. Back to you, Jess. Bye. Hey everybody, what's going on? Episode 125 of Fishtory. The Aquarium PC. At first glance, you see an aquarium, well, hopefully, filled with fish. Look again and you realize it's not just an aquarium, it's a PC. Well, it's an aquarium PC. And the Y2 fish tank chassis combines a beautiful 13-liter aquarium, that's pretty small, with an industrial-looking horizontal PC tower. So it's kind of like the base would be a stand for a fish tank. Well, now it's a PC tower, like a desktop tower. And the company out of China called Metal Fish is making this computer, and they want to actually use the water from the fish tank to cool the computer. I feel like there's all sorts of problems with that. Like, you don't want to run the water through. It could get clogged through small pipes and stuff. But right now, it's got fans set up, and they think that the heat of the computer can regulate the aquarium. They don't have a lot of specifics on this, and so I'm really interested to see if it's just a gimmick or if it's actually something that's viable, where they're going to use the ambient heat from a computer to actually heat an aquarium and to run the life support systems like filtration and things, all off the same electrical stuff as the computer. And then also they're talking about incorporating software so that you can keep up with things like, how your heat is over time, or when you did your last water change, and set reminders and things like that. So should be interesting. If you want to see them, Google Metal Fish Y2 chassis, CHASSIS, fish aquarium desktop. And yeah, I don't know what to think of that. Sounds like a thing that they did. Alright, that's all I've got. This was a short one. I hope you guys have a great day. I'll talk to you next time. Bye. Hey guys, what's going on? It's Alexander Williamson here with Fishtory on the Aquatic Morning Show, featuring Secret History Living in Your Aquarium. That's me. That's my channel. Mine. Copyright 2022. Anyways, I hope y'all are doing well. And today I have a story about the little boy who... there's no moral to today's story, but there is some interesting stuff about today's story, which is they discovered a new group of cells in a group of fish. So in a group of fish in the deep water oceans, they found new cells that are in the eyes, but not just in the eyes. In fact, now they think they've found these cells elsewhere too, like possibly on invertebrates and on other creatures. And what they are is they're visual aids. So they are photoreceptor cells, but instead of being rods and cones like every sort of eyesight we know of invertebrates, this is different. It uses a cell receptor chemical known as rhodopsin, and quite honestly, I don't know a lot about that. I couldn't find a lot about that, but apparently it's a way for them to see fluorescence and inflorescence. So things like fish that would glow in UV light but don't produce their own light, so not not phosphorescent or bioluminescent, but the fish that are UV reactive, kind of like glowfish, I guess. They're able to see just those light wavelengths. It's an extremely narrow band of light that is refracted off of that in the visual spectrum. Most things need to agitate it with light hitting it, and then you see a reflection, and then your brain processes it as light. Well, these little receptor cells in some of these deep water fish are actually able to see that, and they'll see the patterns of UVA and UVB reactive designs. So flowers, for instance, they have a ultraviolet pattern on them that a lot of insects or birds or other animals can then see. And even though it looks like a pretty colorful flower, that's for visual sight-oriented animals with rods and cones. And you'll have your light or dark sensing, and then you'll have your cones. So between cones and rods, one's light and dark sensing, the other one is red, green, blue. In some animals it's red, yellow, black, gray. It just depends, but there are different color, and we know that when they see UV light, they're called tetrachromats, and that's a whole separate thing. But on those flowers, the flowers have evolved with all these other creatures, and it's been useful to them to evolve with markings both in the visual light spectrum, in the infrared light spectrum, and in the ultraviolet light spectrum. And another one is this phosphorescent, or basically it means that it's energized by UV light rather than producing its own light. So any small little bit of light that comes down through the ocean water starts these cells vibrating and having chemical reactions, and then in turn these fish can see them. And we knew that they could see them with sight, but now we're learning that they can actually see the structures that create that light reflection themselves, if that makes sense. So they're seeing some sort of probably gradient, one-tone color of a pattern on that fish. So like if it's a glowfish, they would see all the spots that are colored on the glowfish as bright white maybe, and the rest of their field is tones of gray, like this video. I don't know, we're kind of speculating what they're seeing, but it's kind of interesting. And it's different than how other animals work, like insects and other things may have photoreceptors or photo sensitive cells, and this is different. This is actual sight going through a visual cortex. So it's something new. It's a new way of seeing the world, quite literally, and I think that's pretty cool, even if I don't quite understand all the science of it yet. But maybe one of y'all is smarter than me and can definitely help explain it to me. I'm sure you can. Alright guys, I hope you guys have a wonderful day, and I'll see you on the next episode of Fishtory. Hey everybody, what's going on? We are on episode, 127 now? Whoa, we're getting up there. Okay. Alright, I'm dragging this week, guys, I'm not gonna lie, but I so appreciate you guys being here, and that's why I'm here too. So, today I have a story and it is about looking at the fish that live off of Greenland. And they found that a number of those fish that live off the shores of Greenland... Oh, by the way, Greenland's real cold. It's colder than Iceland. Remember how the Vikings did the switcheroo and they didn't want people to go to Greenland? They wanted people to go to Greenland, not Iceland, so they called it Greenland. That's the theory, that's the old wives' tale. Who knows if that's true or not? Maybe it was actually greener back then. Who knows? Anyways, it's cold now, and it is warming up, but for the meantime, it is icy in the waters off of Greenland, and very icy, to the tune of glaciers and things falling off the continental shelf there, or rather the uprising that is Greenland, the island of Greenland. It is causing glaciers to slide into the ocean. Now, these super densely packed ice chunks, ice cubes basically, like a mile long or miles long in many cases, they're going into the ocean as fresh water, which is cold, and they're going into salt water. Well, the salt water doesn't freeze as quickly it doesn't freeze at the same temperature as fresh water. So the salt water actually allows that ice to make the water colder than freezing. So the fish that live in the water off of there are actually in water that's colder than 0 Celsius, if that makes sense. It's like when you're trying to cool down a drink really quick, if you know that trick, you add a bunch of salt and put ice cubes in salt water brine, and that will cool the water down to like 25 degrees instead of 32 Fahrenheit, 0 Celsius, whatever. Well, it turns out that the fish that live in that habitat, at least 3 or 4 of them, have evolved these antifreeze cells, for lack of a better term. And these cells are a series of enzymes that get into all the different nooks and crannies of their organs. And normally when a complex species or multicellular species is frozen solid, ice crystals form because water expands and crystallizes like a mineral when it freezes. Well, what happens is this totally shreds the cells of like the liver and the kidney and brain, and so it's been a real problem. And sometimes flash freezing has gotten around some of those destructive ice crystals if they can, do dry, basically vacuum sealed dry packing and dry freezing, freeze drying as it's called. That's how we get around it in food. Well, they don't have that luxury, and nature is not that quick, so they're not snap freezing as it's called. They are slowly freezing, and their metabolism slows down, and we knew that. And we knew that there were some chemicals in their body, enzymes, that make it so that the ice doesn't form quite as quickly. It has to be even colder. And usually if you get enough ice on the top of the ocean, then there's this nice warm layer, and by warm I mean like 5 degrees above freezing at best. That's Fahrenheit. So you got some like 38 degree water rather than negative 40 or whatever it is above the ocean up there in the Arctic and Antarctic. Well, it turns out that these critters didn't just have stuff that slowed the freezing of water. It actually breaks up the freezing of water, so it doesn't form crystals. It screws up the stacking and crystallization or mineralization of water. When water is frozen, technically it's considered a mineral. It has a crystalline structure, and what this does is it's an enzyme that gets into any little cracks and crevice, just like a blood cell would or something, but it's probably it is even smaller than that. It's secreted from other cells, and it basically puts a layer on every freezing surface within the body and makes it so those ice crystals can't expand and rupture the cell membrane, which is like the stretchy, gooey outside of cells that we have in our cells and things. It's why when you get frozen, we get frostbite, and then it turns black and the cells are dead and your tissue dies. Well, we knew that goldfish and other fish had these things that allowed them to go into water that's even colder than freezing and not freeze. And that's basically the same trick as having more salt in your blood, or having different chemicals that can delay the freezing effect. But this is different in that it's an actual coating of this antifreeze stuff that stops those crystals from being damaging as they're forming. And so we're looking at this for things like, one, storing food or seeds or plant material if we need to store it cryogenically or frozen for a long time. This may have really interesting implications for that. Also, there's the whole genetically changing different things from plants, crops that can withstand freezes better, to everything else. And what's interesting is that the local Greenlanders eat several of these species of fish that have this same enzyme, and that these are different genus of fish that have this same enzyme. So there's one in particular that has a whole bunch of it, and they're going to be studying that fish in particular with the grant money they've received from this initial study. But the ultimate thing will be, things like space travel, things like preserving people, Walt Disney, and that whole wives' tale too. Cryogenically freezing heads like Futurama. Will you be able to save people's memories and thoughts, completely everything frozen in place without damage to when it's unthawed later? So who knows? Who knows what they'll get from this, but they do think there's a lot of potential. So that is your dose of Fishtory for the week. Thank you so much for listening and supporting. And next week we'll be back on track with a more robust Fishtory and attitude from me, I hope. But I'm hanging in there, just one day at a time. And that's all you can do. Enjoy the beautiful things around you. Stay curious and stay kind. I'll talk to you guys next time.